Hi all, Dr. Clark here again. And today for animal biology, we're going to keep discussing protozoans, but now I'm going to move to the taxonomy of protozoans, at least some of the major groups, so you can understand some of the protozoans that affect human life, or at least have the potential to affect humans, and also to show that connection between animals and protozoans. So I always like to think of things like sperm, you can see here, as that evolutionary advanced protozoan. I mean, if you look, they have a flagellum, and if you looked inside, they'd have, a nucleus that's carrying, half the chromosomes. You're gonna have a few mitochondria. I mean, it's going to be kind of something that doesn't last very long in nature. So the life expectancy is not very long, but it has a single purpose. Okay? And there's a lot of protozoans that we'll talk about. That's exactly their life cycle. Like, they either through asexual reproduction, budding off, or binary fission, or multiple fission, um, they are born and their life cycle is very quick. Now, some last a lot longer in an aquatic system or something like that, but nonetheless, some have a very short life cycle, and it's very particular to a certain region. And we'll talk about some parasites that occur in humans that are very particular to a certain region of our body, and it's that body chemistry and that environment in which they thrive, those protozoans thrive, and nowhere else. And so we'll talk about these because it's important from the standpoint of looking at the evolution of protozoans into the animal kingdom. So in particular, flagellates moving into sponge-like organisms. Okay, so first, we're just going to talk about a few members, few groups of protozoans that can be fairly important. Okay, like Fornicata. This group is important as it holds one of the major parasites that occur in humans. They have a flagella, a feeding groove, so they form food vacuoles, etc. And they are single nucleated, although you'll see that in a couple of examples it looks like they have two nuclei, but it's a bilobe nuclei, so they're connected. And this happens even in our cells. We have bilobe nuclei in things like heterophil cells, which are immune cells. They'll have kind of indentations which make them look like there's two or more nuclei there. And the one that we're most interested in is, well, I guess the one that I'm going to present that's most interesting is Giardia intestinalis. Okay, so Giardia is a genera of protozoans. Intestinalis is the one that we get when we drink water that has this parasite in it, and can get infected with the disease that we call Giardia. And sometimes people call it beaver fever or mountain fever, or lots of names for it. But it's from drinking unfiltered water that has fecal material in it. Typically not human fecal material, but more like sheep or mountain goats or something like that that have defecated in the streams. We drank it. The Giardia intestinalis is in the water system, and then it can go into our system and wreak havoc on us. They feed mainly by flagella, which is going to make a current and push food into the feeding groove. Okay. So here's an example. Okay, a lot of people, when they are looking under the scope and they see like Giardia, they seemingly describe it as a face, like two eyes. Now this is the nuclei. Okay, it's a single nucleus, but it's bilobed. So there's no micronuclei in this organism, which we'll talk about in a little bit. So there's just macronuclei. It's lobed, but there's single nuclei. And then you can see these flagella. These flagella here, there's going to be a feeding groove, like right in this region. Okay, these flagella would create a current and push food into that feeding groove, and then the food would be pushed into a food vacuole. Lysosomes would be released, digested, etc. And so Giardia really can wreak havoc on one's intestines and digestive tract, causing you to your immune system to ramp up and try to fight this organism off, which will cause you, in some cases, to produce a fever, but it will definitely cause you to vomit and have diarrhea and things like that at the same time. Normally, people with Giardia lose quite a bit amount of weight. It's a tough one to get rid of, and in some cases, you really never get rid of Giardia. It can harbor itself in cells, or it can get stuck in like your liver and your potentially your kidney, and it can just go dormant for a long period of time, and then after conditions are right, your immune system is maybe weakened, Giardia outbreak will happen again, and you get it again. So there are medicines for this, so it's not life-threatening. Although in regions where individuals are already malnourished, this is extremely life-threatening. Okay? So if you get diarrhea and vomiting and you're already malnourished, it can definitely cause you to die. And so Giardia probably has quite a few deaths each year contributed to this infection because it occurs all over in the world. Now, maybe not this species, but there's lots of species of Giardia. All right, let's look at another group, Amoebozoa. So the amoeboids, these have a very different body form than Giardia does, in the sense that they have a lopopoda, and the lopopoda is going to be kind of like an arm that they can snake out and create a food vacuole with it, but they can also like pull themselves in directions. Okay, and it's not just a arm; they can move their entire body with these lobopodas. Okay, which are really just extensions of the body that they're snaking out and kind of moving. Okay, some of the interesting pieces have to do with like the mitochondria. So again, we're trying to connect protozoa to Animalia. So we'll look at things like mitochondria, we'll look at their nuclei, we'll look at different functions, their lysosomes, endoplasmic reticulum, and we'll see, well, how close is this group to modern animals? Do they have similar mitochondria as modern animals? Okay, now amoeboids can be either single nucleated or multinucleated. Okay? And we're going to talk about the cyst forming, but they have the ability to go through multiple fission, which basically means that they just divide their nuclei into lots of copies, and they form a cyst, and then they can sit there in a dormant phase for a long period of time, and then when conditions are right, all those cysts, all those new amoeboids can be released into whatever body of water, whatever water column it is, and it's a quick way of mass reproduction. Right. Amoeboid protistas, okay, is an example of an amoeba that is very common to, well, scientific community. You can get amoeba infections from drinking water, so drinking unfiltered water in developing countries that do not have very good water filtration systems you can get this amoeba infection, which will act a little bit like Giardia in the sense that you're going to have diarrhea and maybe vomiting. You might produce a fever because your immune system is attacking this foreign body. And so here you can see a couple things. You can see, these lopopoda, so these long arm-like structures which allow it to move, but it also can allow it to engulf or go through phagocytosis or pinocytosis and engulf things and put them into a food vacuole. Here's a nucleus. Sometimes they have micronuclei, smaller nuclei that can be used as kind of environmental, I guess you could say environmental predictors. So micronuclei typically have the ability to change faster than the nucleus or mutate faster than the nucleus. So when that occurs, it's normally allowing the organism to adapt to a changing environment. It's pretty neat information. Bacteria do this, other protozoans do this, archaea can do this, and it's really a unique little set of genes that allow them to manipulate and change with the changing environment. And this is why, like, bacteria can go through and develop drug resistance really quick. So could amoeboids. So can protozoans. That's why we don't have very many drug treatments for parasites, is they have the capability of resisting drugs fairly quickly. But anyways, it's not a huge player in disease, but it's a very good example of a parasite that can change with a changing environment. Okay. Turkos vaginalis is probably one of the most common vaginal parasites. Okay, Turkos vaginalis is an STD, and so it's a protozoan, but it's an STD that can be passed on. And normally males have no symptoms when they have it, but females will have STD-like symptoms, various discharge and things like that can occur. But again, another protozoan parasite that can be harmful and use humans as a host that, to pass these traits on from, or these organisms on from one organism to the next. Okay, so not going to kill you, but will irritate your body, will cause an immune response, and can be passed sexually from one individual to the next. Euglena, so in lab, most students get to see Euglena. Euglena is one of those interesting protozoans that is kind of like half plant, half animal. And so it has characteristics of plants in the sense that it has chloroplasts. It can go through photosynthesis, but it has characteristics of animals in the sense that it has a flagellum, it has a feeding groove, it can take in food and go through phagocytosis and provide itself with nutrients, etc. It's the whole deal. It's its own kind of package organism where it doesn't need anything else from any other organism. It can create all its own food and do lots of fairly unique things in the sense that it has a photoreceptor which allows it to sense light activity. So during the daytime when it's going to do photosynthesis, Euglena will move up in the water column, do photosynthesis. When the light is low, it'll move away from the surface and start burning up those carbohydrates and in some cases phagocytize and bring in material from outside to help with bodily functions. So fairly unique organism. You'll see that Euglena is often shown in labs because it's a very common genera of protozoan. All right, another one that can affect humans and have a fairly unique life cycle is Trypanosoma, and there are lots of different species of Trypanosoma. Brucellii is the one that, or Brucei is the one that is probably the most common in the case of humans, and that is the one that causes what we call African sleeping sickness. And so just like malaria, which we'll talk about in a second, okay, it's a protozoan. It's passed on by the bite of an insect. In this case, mainly the one that affects humans is a tsetse fly that bites a human, passes trypanosoma from the salivary glands into the blood system of whatever mammal it is going to infect. In the case of humans, it gets in, our immune system builds up a response. Okay, these trypanosoma get phagocytized, your immune system, your heterophils, okay, your big white blood cells will eat it, present it, but this species has a very unique evolutionary trend, and that is that after it's been digested and things like that, somehow either chemicals flow throughout the blood system, but the majority of trypanosoma that have not been consumed and not been damaged by the immune system, they switch their outer protein coating. So now they're not detected by the immune system. And so this will happen over and over again. And so trypanosoma is very difficult to get rid of out of the bloodstream once an individual has it. There are medicines for this. It can cause individuals to die. Basically, trypanosoma just eats your red blood cells, and it'll even eat some white blood cells, and it'll cause you to become anemic, etc., and eventually die if you're not treated. There are treatments for this, like I said, but the other thing that's interesting to scientists is this ability to change protein coding. We're interested in this from quite a few point of views. Now, let's say we're going to transplant an individual's a new organ or something like that. Well, we know the immune system could attack that and destroy it. But what if the outside coating of the new organ had the ability to switch and reject our immune system? So it's not going to be affected by the immune system. So maybe, the immune system reacts to it and starts deteriorating it maybe initially, and then it switches its coat and the immune system doesn't react to it ever again. Okay? These kind of ideas, maybe from a medicine point of view, organ transplant, blood transfusion, lots of different things, because other organisms have these capabilities of switching their protein coding, we're interested in how that works. Like, what's the cues and how quickly can it be done? In the case of trypanosoma, it's fairly fast. So flagellates, like trypanosoma and the next one that we're going to talk about, malaria, have kind of this unique body form in which they have a free flagellum, okay, but it's an interesting kind of setup because the whole body is similar to a flagellum, and it has this ability to fold its membranes and create feeding vessels or feeding vacuoles, food vacuoles, by folding its membranes over, but it also has the ability to swim and to move and be mobile through a semi-aquatic or aquatic environment. Other things that are interesting, the mitochondria itself has a very unique kind of setup, and the nucleus is fairly similar to animal nuclei, and it just shows a lot of that connection between primitive protozoans, more advanced protozoans, and the animal kingdom. So here's malaria, again, flagellate parasite. Most people know it's passed on by mosquitoes, certain species of mosquitoes. Okay, now there are quite a few different types of malaria, so we're not just talking about single types of malaria, but malaria passes on sporocytes through the saliva of typically a mosquito, um, and then those sporocytes go into the liver, they mature in the liver, okay, and then they burst out of the liver, enter the bloodstream, okay, destroy red blood cells, and they just keep going on. Now, again, like I said before, when we were talking about parasites, parasites will often have intermediate and determinate hosts. In the case of malaria, it's interesting. The life cycles of malaria treat our body, treat different regions of our body like different hosts. So like our liver is like an intermediate host. And so when the sporocytes get into our liver, they reproduce asexually, okay, mature in the liver, and then rupture out and move into the bloodstream. When they get into the bloodstream, same kind of system. They're immature, they become mature in the bloodstream, eat hemoglobin, rupture the red blood cells, and then they get back out into the bloodstream and they can start reproducing sexually. So it's interesting. We'll look at other parasites that have multiple stages, intermediate hosts and definitive hosts. But in the case of things like malaria, we're both the intermediate and definitive host. It just depends on what part of the body that organism or that stage is in. Okay? But malaria is a very serious disease in the world. It kills roughly 1 million people every year. Okay? We have anti-malarial drugs. They're very expensive. And so also when you use anti-malarial drugs, you build up resistant malarial strands that are resistant to your antibiotics or your malarial drugs. And so now there's quite a few malarial drugs that are completely useless. And so we see this functionality of this endless cycle of evolution. There are currently groups of individuals that are trying to rid the world of malaria in different ways using CRISPR. And this technology will allow for us to either develop mosquitoes that cannot pass on the sporocytes. Right? That's one method at which some individuals are using, like at Berkeley. And then there's another method in which we would eliminate all males from a population. So there are no males, so therefore the mosquitoes cannot reproduce, and the mosquitoes will go extinct in that region. You know, in my opinion, the Berkeley method is better than the European method, which would make the mosquitoes go extinct. However, either method will work most likely and will reduce the amount of people that die from a malaria infection yearly. Okay? And so you probably will see this in the next coming 5-10 years. You'll probably see this technology at least coming up for regions that accept it and want to see a reduction in malaria in those regions, mainly Africa, but also India and other parts of the world that are tropic. That's where malaria is most rampant. All right, so we talked about flagellates, we talked about amoeboids. Now we need to talk about another group that are kind of more complex, and that would be the ciliates. So instead of having one flagellum or a couple flagellum, now they have a bunch of miniature flagellum, which we call cilia. Ciliates have that unique system in which they have cilia, but they also have macronuclei and micronuclei. So they have this capability, majority of them have this capability of passing on micronuclei. So prior to this, majority of protozoans, if they had sexual reproduction, it wasn't as diverse and wasn't as complex as, say, the ciliate sexual reproduction. ciliate sexual reproduction is probably what gave rise to sperm and egg. Right? Is probably what gave rise to that sexual or sex being developed in organisms, because they have the capability, ciliate protozoans have the capability of doing what we call conjugation. They can exchange micronuclei. So they can come together, just merge their membranes together and pass little chunks of DNA back and forth. Right? And only organisms that are different from each other can do this. So there's no, like, you can't go through conjugation with your clone. And so this is how we think, is how scientists think sex developed, is through this conjugation method, and then eventually one sex became really small, like sperm, and one sex became really big, like egg, and that's the way it is. Like, there's no real in between, and so we had this divergent evolution when the intermediate sizes were eliminated because they weren't as good at swimming, or they weren't as good at providing nutrition. So here you can see this is Ciliphoria. Paramecium is a good example of this group, which most of you have probably seen in pond samples or whatnot. Okay, they often have a feeding groove or an oral groove, which will make food vacuoles, and those food vacuoles then get lysosomes or digestive enzymes get processed and put into them so they can digest whatever the food was. They move with cilia. They have a macronuclei, typically just one, and then they can have multiple micronuclei, which are those small regions which is going to have a subset of DNA. You can kind of think of micronuclei, if you know much about bacteria. Bacteria have no nucleus, okay, but they have a nucleoid region which houses their single chromosome, and then they'll often have little regions which we call plasmids, which house little chunks of DNA. And you can think of micronuclei as just kind of the protozoan version of plasmid. Okay? And so that capability really allows them to produce in multiple ways. So they can produce asexual. They can produce by binary fission, either on the transverse method or horizontal fission. Okay, so they can divide. It depends on the organism how they divide, but they can divide in many different ways. Okay, they can also bud occasionally. It depends on the species again. Right? But interestingly is the sexual part. The sexual reproduction is most interesting in ciliates because they go through this conjugation. Okay, and so here is just showing you paramecium going through conjugation. So it's representing two unique individuals. Now the macronuclei is not that important because that's not being passed on. It's not being migrated, it's not being manipulated, anything like that. But it is showing that there are two distinct individuals because the color of the micronucleus is different. Okay? One's red, one's yellow. Okay? So the organisms would then copy their micronuclei, and they might copy their micronuclei many, many times. Okay? And then they'll pass those micronuclei in between each other. And so, in some cases, it can be recombinated. So you can pass it over and it gets mixed, and then they pass another one back. So there's lots of ways at which this can occur. You can get micronuclei manipulation. You can get micronuclei multiplication. So you just keep on making more and more micronuclei, and you pass those on. You can even get macronucleus going through reproduction and making new macronuclei. And some organisms have the capability of first going through micronuclei exchange and then go through basically meiosis and divide the cells and utilize the division of the cells to create 4 new individuals. So first you'd have this conjugation where you're exchanging material, and then the individual breaks off from conjugation, goes X-conjugation. Okay? Then that individual goes through basically meiosis and starts dividing itself. But it's because the micronuclei are not all the same because you went through conjugation, the individuals that come out are distinct genetically speaking. Okay? And so this is probably the precursor to the meiosis that we know when we start looking at the formation of sperm and egg in animals. Okay? So there's a lot of things that we don't know though, right? Because protozoa are very numerous and very diverse, and you have animal-like protozoa, and you have plant-like protozoa, and you have animal and plant-like protozoa. You have very unique systems, right? And they have very distinct ancestral lines. Okay? So amoebas and channel flagellates do not share the same ancestor. At least we haven't found it. Okay? So this makes it very difficult to get that monophyletic grouping. And so right now it's a polyphyletic group, and there's at least 6 different groupings. Okay? And so maybe in the near future you might see that the Protista group is broken up into 6 kingdoms instead of just 1 kingdom that we lump everything and call it Protista. Now the other piece of this is, there really is very little fossil record. Okay? You have to think about protozoa and protista. They just don't fossilize like other organisms. Okay? There's really no hard body parts. So, to get them to turn into rock, you have to have like perfect conditions in order for you to get an imprint of one of these. And they're microscopic. So, you probably wouldn't even know it was there unless you're looking for that under a microscope, trying to find, protozoa indentations in some kind of sediment rock. Ribosomal RNA is not complete. It's not giving us the best picture. There are, supergroups that come out in ribosomal RNA. Now, is this because we have not found the ancestral groups? Maybe. Maybe they still exist. Is it because all the ancestral groups are extinct and, that these protozoans are really not connected to each other? That's possible too. Right now, I think what I can say about the Protista protozoan grouping is that the reason why we have these supergroups and we can't figure out, and we have these polyphyletic connections, is we don't have enough information. We've not studied it enough. Right? We have a lot of holes and missing information. So in the future, when scientists get around to it, we'll probably have a better idea of the grouping or the group that we call Protista. So here you can see this is based on ribosomal RNA, 18S ribosomal RNA. You can see like things like the ciliates, the paramecium. Okay, they come out not related to things like the entomoeboids. Okay, these guys don't even have a mitochondria. So this is both ribosomal RNA and based on mitochondrial differences. You have other things like Trypanosoma, which is not related. It has its own ancestral line compared to things like Giardia. So you have a lot of things like Malaria, Plasmodium is linked to Paramecium, but it is not linked to things like Trypanosoma. So you have a lot of very different kind of groupings that fall into what we call Protista. And we really just lump them all together, even though we know that animals have a single line, fungi have a single line, plants have a single line, But then when we get into what we call Protista, we just jumble it all together, slam it all together because it's kind of a catch-all, and so it's a problem that more research will eventually fix. Okay? All right, so with that, we're going to move into talking about the first group of animals, Porifera. Okay? So next time, we're going to talk about the connection between protozoans and Porifera.